So here we evaluate a new ultrasensitive ctDNA test. So this is a cohort of patients with triple negative breast cancer treated with neoadjuvant treatment. So we have a blood sample before the treatment, at the end of neoadjuvant treatment, during the neoadjuvant therapy. So we observe that with an ultrasensitive ctDNA test, is an ex-personal assay. At baseline, we were able to detect 100% of lesions. During treatment, the majority of samples are below the ultrasensitive range, so below 100 ppm. Interesting, so generally in breast cancer, we use a pathological complete response, the PCR, as a surrogate marker, as a prognostic factor to guide the adjuvant treatment. So, and we can see patients we achieve the PCR have no relapse, while patients with no PCR, we have 14% of relapse. If we use now the ctDNA detection after neoadjuvant treatment, we can see that patients with ctDNA undetected test after neoadjuvant treatment, we have 3% of relapse. So very, very few relapse. When patient is a positive test, we have 60% of it. And interestingly, as a breast oncologist, as I guide my adjuvant treatment based on this PCR response, we can improve the prognostication of this patient because this group, if we measure the ctDNA, you have here the ctDNA positive with no PCR and the ctDNA negative with no PCR. And you can see, in fact, the huge difference in terms of relapse. And for me, which is very interesting, is that patients with no PCR and ctDNA undetected, we have a very, very few relapse rate, 5%, which is compatible with treatment de-escalation. I can't think about treatment de-escalation if I have 5% of relapse. Why? I can't do treatment de-escalation if my relapse rate is 20%, like we observed with third generation ctDNA assay. And this is confirmed here. ctDNA detection matters, either if it's below the 100 ppm, the ultrasensitive range, or above. This is the same relapse rates. So yeah, for me, these data are really promising and were confirmed yesterday in another cohort, so which I think is very important to have two different cohorts showing the same results. So based on this, as a medical oncologist, I want to design a clinical trial based on treatment de-escalation in this subgroup of patients is no PCR, but ctDNA undetected.